In this chapter, we begin our exploration of simple plants or plants in general. And in this case, we're going to be looking at specific plants that are, we refer to them as seedless plants because they don't produce seeds. The seed-producing plants are the ones to later evolve, and we would refer to them as the more complex plants, where we're going to be looking at the more simple plants in this chapter. So looking at section 1, which is titled The Origin of Land Plants, your learning objectives include to explain the relationship between the different algae clades and plants, and then we want to describe the haplodiplontic life cycle, which we already actually saw with brown algae in the prior chapter on Protists, and then distinguish between the Sporophyte and the Gametophyte, and these are generations. The plant alternates between a diploid and a haploid body, plant body. We'll see that in a bit in this section. And then identify 2 major environmental changes for land plants and what those adaptations would have to be. So in other words, we're talking about the ancestors of land plants were in the water, and then they moved on to land. What were adaptations necessary for that? So all green plants or green algae and the land plants, they do share a common ancestor, which is going to date back to a little over 1 billion years ago. And so we're talking about this point here on the cladogram. So we say this is present time here. We went back in time. This would be where that common ancestor would be. And this is supported by analysis of DNA, which is molecular data, and structural features as well. Now, collectively, we're going to know these this group as green plants overall, and then there is the land plants. And part of the green plants overall will include a couple of groups of algae, which would be the green algae. So all of these guys right here, we might refer to as green plants in some classification schemes. And then we don't get into land plants till we get about right there with the liverworts, about right here. And then we move on to the right. And keep in mind, we're going up this cladogram here. That means we're progressing forward in time until we find these common ancestors. Now, not all photoautotrophs are plants. We know from prior photoautotrophs, there's the cyanobacteria, which are prokaryotes, brown algae, right? And the red algae. You see the red algae out here would be a group that shares some ancestry with what we would refer to as the green plants. And they are photoautotrophic. They use sunlight to make their own food. So life on land, you got to have overcome many issues. One of them is when you come onto land, you're going to lose water quickly. If you live in water and then you take that organism out of the water, they likely don't have adaptations to prevent good adaptations to prevent water loss across their body. And plants have a body too. If you look at it at a tree, the leaves, the body of the leaf, the body of the plant. So they need that protection from water loss, and then they also need protection from the harmful effects of the sun, specifically ultraviolet light. And so those are a couple of issues that the plants need to deal with, because when you're in the water does provide some protection from that radiation from the sun. Colonization of land also required the help of an association, a symbiotic association with fungi. And these fungi help make nutrients available to plants. And I saw this picture online. I thought I would share it with you. You can see down here, these types of fungi are called myco, which means fungus, rhiza, or mycorrhizae, plural. Mycorrhizae is the name given to these fungi that have an association with plant roots. And you can see here, we have 2 of the same kind of plant, and one of them it has mycorrhizal fungi growing with it, and the other one does not. And you can see that the one on the right does not grow as robust or as good as the one with the mycorrhizae. So many, or a lot of land plants, most land plants are going to have these kinds of symbiotic relationships with fungi. So the evolution of land plants includes the story of their origins going back to where green algae and land plants share this common ancestor. And so the green algae themselves, you look at this cladogram here, there's two major clades within the green algae, and this is seen on the right here. So you have this green algae right here and this one right here. There's the chlorophytes and the charophytes. And the charophytes have characteristics, both molecular and physical, that strongly suggest that these guys right here, charophytes, they weren't covered well in the last chapter. We talked more about the chlorophytes, but the charophytes do exist. And the molecular data shows that these guys share the most recent common ancestor with our land plants. So you can see the land plants start right here with the liverworts, the label there for the land plants in this group, which starts about right there and goes this way. Okay. Looking at land plants, some characteristics for them generally. They are multicellular in two major stages of their life cycle. They're going to have a multicellular plant body, which is an entire plant all on its own, and then a multicellular diploid. And we'll see the alternation of generations general, and then we'll see it over and over again as we look at specific life cycles of plants. So there's going to be a trend, though, when we go from the more primitive land plants, like the liverworts and the mosses and the hornworts, and then we start moving to higher and higher plants. We're going to see a trend towards a diploid embryo protection. So we're going to see the embryo is what you get when the sperm fertilizes the egg, and it's now diploid because sperm and egg are haploid, and the parent plant is going to protect that embryo from drying out, from desiccation. We're also going to see a trend towards smaller haploid stage. So with the more primitive plants, like the Bryophytes out here, which are the mosses and this group right here, the haploid stage is the more dominant one. It's the one that seems to in appearance to be more ecologically important. It's the one that does photosynthesis. Generally, it's a larger plant body than the alternate diploid stage. Okay, so that's what that's meant. And so when we see a trend here, for the mosses, the haploid stage is dominant, and then as we move this way to the higher plants, we start to see that the haploid no longer dominates and the diploid stage now dominates. So this is that same cladogram, just looking at the relationships here. We go back here to find a common ancestor between your green algae and the rest of the plants here, and collectively we would call all of these guys, according to the diagram up there, they're all the green plants. The red algae would not be considered there, and there would be a common ancestor for those to all of the green plants. Then we would have to come up here to find the split between the more advanced green algae and then the rest of the land plants. So this would be your common ancestor right there for that. So looking at the adaptations for terrestrial life, first of all, we got to prevent water loss across the body of the plant. And part of the body includes things like structures like leaves and stems. These structures are going to have a waterproof coating or a cuticle, waxy cuticle. Wax is a kind of a lipid. Remember lipids from your chemistry, the lipids don't mix well with water or polar things. So if you have a coating of a lipid on the outside, it's hard for water to get through and evaporate away from the body. The leaves will also have stomata, which you probably talked about when you covered photosynthesis. Stomata are going to be the little openings that are on the epidermis, for example, under the leaves of plants, on the bottom surface of the plant leaf. The stomata are there where the plant can release oxygen and take in carbon dioxide for photosynthesis, but water also leaves through the stomata, and when times are hard and the plant is water stressed, they can close the stomata to prevent excess water loss. And then plants have, eventually they evolve, for land plants, they evolve these tracheids, tracheids is a special kind of tissue or a special kind of cell type that's found in water-conducting tissue. That water-conducting tissue is called xylem. And together, xylem and phloem are referred to as vascular tissue. And generally speaking, the vascular tissue is how water is conducted up and down the plant. The xylem, like you see in the diagram right here, conducts water up the plant body, and phloem basically from the leaves back to the rest of the body, which means back down the plant. Now, in dealing with the move on to land, the plant also had to evolve some sort of mechanism for countering the damaging effects of ultraviolet light. That's the same radiation that gives you a sunburn. And in this case, the shift to a dominant diploid generation. You remember the difference between 1N, which is haploid, and 2N. And we'll see here that the plants alternate between these two generations. But for the earlier plants, the more primitive plants that are tied more to water, when you move on to land, you're going to be exposed a lot more to direct sunlight. You need a way to counter the harmful effects because ultraviolet can damage DNA. So if you have only one copy and sunlight damages a gene on one of those chromosomes, now you have a bad copy and then you don't have anything else to help out. But if you have two copies, ultraviolet could damage one, but you still have one good copy. So that's sort of an idea of the mechanism or sort of a partial, at least, explanation as to why maybe the diploid generation is the one that evolved to be more dominant. Plants have what's called a haplodiploid, which means haploid and diploid life cycle. So they alternate between a multicellular haploid and a multicellular diploid. For comparison, humans and other animals, most other animals are going to be diplontic. We only have a multicellular diploid. Our bodies are diploid. We do produce haploid, but a sperm and an egg, which are haploid, don't go turn into a whole other body that's walking around with all haploid cells. Instead, the sperm automatically fertilizes the egg, and you get the next diploid generation. For plants, they produce haploid spores that grow into an entire haploid body. And then that haploid body then produces gametes that get fertilized and then produces the diploid generation. So they alternate between these two multicellular stages. Animals do not do that. So this next section here does talk about that haplodiplontic life cycle. And the two stages. Okay, the diploid stage is going to be referred to as the sporophyte. And that you need to know, that's a vocabulary word. And that's where all of the cells in the body of that plant are diploid, okay, overall. Now, the sporophyte gets its name because it's going to be the plant generation that produces spores. So if all of the cells are diploid in that body and you're going to produce spores for the haploid generation, then that has to be meiosis. So some of the cells are specialized in the sporophyte plant that are going to go through meiosis. This is similar to in the gonads in animals where meiosis occurs to produce gametes. But here, meiosis is not going to produce gametes. Meiosis is going to produce spores. So that's something that you need to make sure you contrast between plants and animals. Meiosis in plants produces spores. Meiosis in animals produces gametes that end up combining and fertilizing right away. Now, what are the special cells found in the sporocyte that go through meiosis? They're called spore mother cells. That's one vocabulary word, but that's also equal to saying sporocyte. So be familiar with both terms because either one of those terms are going to end up in diagrams and questions. So sporocyte and spore mother cells, those are the special cells that are diploid and go through those two divisions of meiosis. First is the reduction division, producing haploid cells. And then those two go through a regular division that just separates the sister chromatids, producing, as was discussed in the prior chapter, for sexual reproduction, producing these haploid cells, and they're all unique genetically because of crossing over and the random alignment of the chromosome pairs. The thing here with plants is that these are not going to be gametes. These are going to end up being haploid spores. Okay. Now, you're going to get 4 haploid spores, and those spores are the first cell of the Gametophyte generation. So these cells are going to be spread in the environment. They go and they land somewhere, and they germinate, and they grow by mitosis to produce the Gametophyte plant. Okay, so that's the next generation. It's the haploid stage, and the Gametophyte is the name of that stage, and it's 1N. Now, Gametophyte is going to be the one, it gets its name because it's going to be the one that produces gametes. And you're already haploid. All of the cells in this body are already haploid, so it's going to be mitosis that has to make these, but not just mitosis. It's going to be mitosis, and those cells flip on a different switch. They behave differently. They're not going to behave like the cells that produce them. And when cells flip on a different switch, we learned that that's called differentiation. It's just a matter of turning on a different set of genes. All of the chromosomes are identical in the entire body, but you turn on a different set of genes that programs that cell to behave like gametes, right? So here you produce your gametes, and then they're called sperm and egg, and the sperm and egg, they fuse together in a process called fertilization, and that'll give you your diploid zygote, which is going to give you, eventually grow by mitosis to give you the sporophyte generation. That should be more clear right here. On the top part of the diagram, we have our gametophyte plant. This is represented by an oval structure. So these things look, in some cases, very much plant-like. When we cover mosses, the gametophyte is going to be a little multicellular plant. They're generally small plants, these mosses, but it's going to look like a plant. That oval represents that plant. So that plant would be the gametophyte. And every cell in the body of that plant is haploid. Okay? On the other side here, this is the diploid side. This is the sporophyte. And this will look something like a plant as well. It's going to be a multicellular plant body, and all of the cells in there are diploid. And so this is going to be a diploid stage. Okay? And so we start on the prior slide, we started talking about the sporophyte in the first part. So let's just start with the sporophyte here. So here's your sporophyte here. Some special group of cells or an organ in the plant, which this term is not on that prior slide, but they're called sporangia, and sporangium would be, it would end in IUM for singular. The sporangium is where the spores would be produced. It's in the sporangium. It's an organ within the plant. Plants have organs just like animals have organs. So this is a special organ made of diploid cells, and within there, that's where you're going to find your spore mother cells, which are also called sporocytes. Okay, it's those special diploid cells, because remember, all the cells in this part of the plant are also diploid, just like the rest of the plant. Those cells go through meiosis, and when they go through meiosis, each one of those spore mother cells, or sporocytes, produces 4 cells. Each one of those is the first cell of the next generation. These spores get dispersed to other places. They land, they go through mitosis and differentiation to form your next generation gametophyte. Now your gametophyte, within there, the gametophyte is going to have special organ structures, which were not labeled in here. These organ structures are not called sporangium, like over here, they're called gametangium or gametangia in plural form, gametangia. Gametangia is going to be, and the cells of the gametangia, so the gametangia is an organ within the gametophyte. Those cells are haploid, just like the rest of the plant body, and those cells have to go through mitosis. Okay? So the cells of the gametangia go through mitosis and differentiation to produce egg and sperm. Okay? And then the sperm can fertilize the egg, restoring your diploid, giving you the first cell of the next generation. It's called zygote. We know that from animal biology, from studying animal reproductive system. And then the cell goes through mitosis, development, embryo, as the embryo may be become dormant for a while until times get good, like say if it's in a seed, when we cover seed plants, which is next chapter. But the embryo, this is going to be the one that's protected for a while, and it's the next generation is just there ready to grow. If conditions are good, it's going the cells of the embryo is already multicellular at this point. It's just they're being protected by either the plant or a structure the plant produced. And this embryo is just there waiting to germinate and to begin to grow again. And when it does, multiple mitosis and differentiation to produce your full body of the next generation sporophyte. If we compare that to a fern life cycle here on the right, you have your sporophyte generation on the bottom. And this is your gametophyte generation. And this is your gametophyte right here. This is your multicellular plant. They're kind of small though when it comes to ferns. This one is about half a a centimeter in size and diameter, but it's multicellular, and on there it has those special gametangia. It has the ones that produce the eggs and the sperm. The sperm end up fertilizing the egg. The fertilized egg is the zygote over here, is now diploid. Remember, these are haploid cells over here. That zygote begins to grow. Here's the embryo. It's being protected by the prior generation plant body. Remember, embryo protection is important to keep from drying up. As the embryo begins to grow, there's your sporophyte. The gametophyte is there, still protecting and helping it for a little bit until it gets a good hold. The cells in this one are diploid. This is the gametophyte, and the cells there are still haploid because that's this plant body right here. Eventually, the gametophyte degenerates and dies, and your next generation sporophyte grows into your fern plant. The fern plant will have sporangia growing from the bottom of the leaves, and in the sporangia, that's where meiosis would occur. Meiosis produces spores, which is the first cell of the next gametophyte generation. Those cells land somewhere, germinate, and then grow into your next generation gametophyte. So this is comparing a specific life cycle to the generalized alternation of generations. So the fern alternates between these generations. So the haplodiplotic characteristic of these land plants. We don't see this in algae. At least in green algae, you don't really see it. You might see it occurring in other groups of algae which are not in this clade, like brown algae. They're in a different clade. They're in the chromalveolata. Remember, this is the Archaeplastida group from our study of eukaryotes in the Protist chapter. But here earlier I mentioned that as you move from the Bryophytes, which are the mosses, the primitive plants, and they're still alive today, to the most advanced plants, the ones that evolved most recently, the flowering plants. These are the angiosperms here, like this tree here. We start to see that the gametophyte becomes larger. For a moss, this is, for the more primitive plants, let me back up. Below the line here is the gametophyte. So this is gametophyte, above is what the sporophyte looks like. So down here is your gametophyte, and for the primitive plants, the gametophyte is more dominant. That's why the blue line is brought here. As we go through evolution and time, we move forward from the earliest plants when they evolve, you move forward in time. You come down here, and it seems that the gametophyte gets reduced to just a small number of cells, and that's it. Whereas the sporophyte generation dominates. So you get things like these familiar trees. Here's that fern. This is that little 0.5 centimeter little gametophyte for the fern. For the mosses, they're very small plants, but the gametophyte dominates and the sporophyte totally depends on the gametophyte. So that's what this is talking about. We see a trend as the land plants evolve from going way back when the earliest plants that probably resembled our mosses today. You're going to see a trend in the reduction of the gametophyte. The gametophyte generation starts off being dominant and then becomes just a few cells in size. But the life cycle, general life cycle, can still be connected to that. So that's the big idea there. And you'll see this when we look at the specific life cycles of mosses. You'll see how the gametophyte generation is dominant. Sporophyte totally depends on the gametophyte. It reverses as you go through the sequence from these primitive, early evolving land plants to the more advanced, later evolved plants, like the flowering plants. You see that it's the sporophyte that becomes dominant. Okay, so that's what this part of the slide is talking about. The mosses are on the left side, the angiosperms are on the right, and this is a progression to more and more advanced plants. And what we see in the fossil record as the earliest plants to appear, the more and more evolving more characteristics that are adapted to land. And this is part of that, adapting with that diploid, 2 sets of chromosomes. That's better protection against the damaging ultraviolet. So for the mosses, the more primitive plants, the gametophyte is dominant, and it depends. The sporophyte is dependent on it. So the sporophyte, which is right here, depends on the gametophyte. For the angiosperms, the gametophyte becomes so small. There's just a few cells. You can see this little a little pollen grain. These are pollen grains. This is what is going to become the seed. That's your gametophyte generation. Is very, very small compared to the large sporophyte, which is like an entire tree. And so that's what we're looking at when we say there is a trend towards a reduction in the gametophyte generation in the evolution of land plants. In this section, we're going to look at bryophytes. The bryophytes is a group that includes the mosses, the hornworts, and the liverworts. And generally, we refer to these as seedless. Seeds haven't evolved yet with this group. Again, these plants are still alive today, but we go back to find common ancestors that probably evolved these characteristics. So no seeds and no vascular tissue. So these are seedless avascular plants. And your learning objective here for the bryophytes, which a major thing to know is that the dominant generation is a gametophyte generation, is your objective is to describe the adaptations of bryophytes for life on land. So what are some things that allow them to move on to land? These bryophytes, if we go back to a more simplified cladogram here, the bryophytes include the liverworts, the mosses, and the hornworts. And these right here would be share the you go back and you find a common ancestor to them and other the land plants. And so these guys here are descendants from some of the earliest land plants, is the point trying to be made here. In other words, so when I say that they're more primitive, is that they probably resemble something similar to what those early plants that began a lifestyle on land. They got the characteristics for that. They're called non-tracheophytes as well. That's another way of saying not vascular, because in order to have vascular tissue, you have to have those tracheids. Those tracheids are that special tissue within xylem made of these cells called tracheids. And so they don't have tracheids or other conducting cells. And so for this reason, they can't get water very high up above the ground, and this is going to limit their size overall as a plant. They can't get very big because they can't get water efficiently up to higher parts above the land. And then in the last section, I mentioned mycorrhizae. So mycorrhizae is the name of a fungus that has a symbiotic relationship. That's what this association is with plants. So these mycorrhizal associations, that's another way of saying the mycorrhizae. They form a relationship with the structures that are found underground. In the case of mosses, they're not true roots, but root-like structures that are going to help these plants obtain things more efficiently from the ground, like water and minerals and plant nutrients. So again, it is a symbiotic relationship between fungi and plants. So the basic bryophyte characteristics, they are adapted to terrestrial environments overall, so they'll have some protection against completely drying up. They're not fully adapted onto land. They still are going to require water for completing their life cycle, and that's down here. They just haven't evolved all of the tools for it. When we look at animals, the ultimate move on to land was when they protect the embryo from drying up in an amniotic egg. Here, these guys still need water because their sperm need to swim to fertilize the eggs. We're going to see later on in more advanced plants, the sperm is packed inside a pollen grain, and the pollen gets transferred to the female by way of wind. And then the sperm inside the pollen can then fertilize the egg, so no water is needed there. So some similar things, some similar analogous trends, not homologous. There's about 16,000 species of bryophytes, we call them in general, and they do include a group called the Bryophyta. So bryophytes is just kind of a catch-all for all of these that are non-tracheophyte or avascular. The three major clades, there's the liverworts, and they're grouped collectively into a formal taxon called the Hepatophyta or Hepaticophyta. They're synonymous. The mosses belong to a phylum, and it's a clade, so its classification matches its monophyletic would be the Bryophyta, and then the hornworts belong to another group called the Anthocerophyta. We'll see those spelled on the title slides coming up. So your Gametophyte here is the dominant one. It's the conspicuous one. Generally is photosynthetic, though we always see exceptions to these rules. The Sporophyte here is dependent on the Gametophyte. It's going to be smaller and depends on the dominant Gametophyte generation for its development and growth and nourishment overall. So the total dependence on the dominant Gametophyte generation here. So the first group we're going to look at is the liverworts, and this translates into a word is a plant. And liverwort gets its name because the body, which you can barely see down here, but the body lays for what gives this group its common name, has a flat body that occurs in lobes like a liver. Your liver inside your body has different sections to it, or lobes. And so whoever was naming this said, hey, that kind of looks like an animal liver. And it turns out that most mosses don't even have this sort of body form to it. They're more like mosses that grow up away from the ground. Now, this structure that you see right here is actually a female reproductive structure. On there is where, and you're gonna study this in more detail in the laboratory part here. But up in there, this is the Gametophyte. It's the dominant generation, the Gametophyte. In the Gametophyte, and these are the females, they look like palm trees. Under there is, and since this is a Gametophyte, it's haploid. So I spelled Gametophyte, believe it or not. So in the Gametophyte, let me spell it. All the cells that make this up are haploid, and under there are the special organs that are called gametangia. And since this is female, the specific name of this one's going to have a specific name. There's a difference. Generally, when you have one that produces eggs, it's a female gametangium. And so let's go ahead and name those here for your notes. The gametangia, remember that's plural for gametangium. What is built in there? The gametes. And this is the Gametophyte, so the cells are already haploid, so you cannot do meiosis like you do in animals. So they're called gametangia, that's plural. And the female is called the archegonium, and it's archegonia plural. So this is the female gametangium. The male gametangium, which produces the sperm, is called the antheridium. And if you're following with how we pluralize these using Latin rules, then antheridia would be plural. This is the male one, and that produces a sperm. This right here is a female. The male, it looks different. The male is going to look, it's going to grow up on a stalk just like that for liverworts away from the plant body. But instead of having little finger-like projections facing down, it's going to be more almost like a semi-cup type of structure in there. And then within here would be the antheridia, and they require water. So let's say rain or dew starts dripping down from a structure above, maybe a plant growing above, and a droplet of water lands in here. That's going to cause the sperm to splash away to a neighboring plant that may be female, and the sperm lands on the female. And then because there's water's around, maybe in the early morning, there's a lot of dew, and the water accumulates on the plant. The sperm can then swim underneath and go fertilize the egg. And the thing about land plants is they retain the embryo, at least for a little while, and they protect it. So the fertilized egg is the zygote then divides, becomes the embryo, and the embryo starts growing out of the gametophyte. So this gametophyte is the dominant generation. You're going to see the life cycle in your lab manual in more detail. Okay, you want to make sure you relate it to your generalized life cycle. Like I said, most of them actually look like mosses rather than this flat body of the liverworts. And you're going to have to know this name for the laboratory. And I might use it during a lecture exam, so get familiar with it for both. It belongs to the formal taxon at a rank of phylum. It is the phylum Hepatocophyta, and some resources will use a shorter version, Hepatophyta. And in an earlier lecture, I mentioned that the word phyto, which is the root of this, phytos means plant. And I think the origin is Greek. Oops, it means plant, but the origin is Greek. So it's a Greek origin. Okay. And so the hepato, like hepatitis, hepato refers to liver, so literally translates to liver-looking plant. And then you have the mosses, which belong to another phylum, a formal taxon called the Bryophyta or the Bryophyta. And there's that root again that means plant, right? So here, the Gametophytes, they're gonna be small and they look a lot more like plants to us. I'm gonna circle one here in the picture there. That's the body of one, and that's your Gametophyte. All the cells that make it up are Gametophyte. Now remember, these guys do not have vascular tissue. These are avascular plants. They have no tracheates. And in order to have true leaves, stems, and roots, you have to have vascular tissue. So while these look, it looks like little leaves on the plant, that's your they're not true leaves or stems or anything like that. So they have a stem-like structure, they have leaf-like structures, and they have root-like structures called rhizoids, which just means little root, but they're not true roots. And those rhizoids help anchor, and they draw up water, but they don't fit the definition of an organ of a root. Like a stem or a leaf requires vascular tissue within there. I mentioned this term earlier. You see it now being played out in the notes. Gametangius, plural for gamete, gametangium. And the gametangia is an organ found within your gametophyte plant. Okay, and this is where you're going to have two kinds. You're going to have the female, which is the archegonia. Archegonium is singular, and this would produce the eggs. The antheridia produces the sperm, and they have flagella, and this is why you need water, so that the sperm can swim to the egg. So on the left is an image showing what these plants look like, and they're actually showing both. There on the bottom is the gametophyte that has the rhizoids that anchor into the ground, and growing out of it used to be an egg that got fertilized and then turned it into the sporophyte generation. The sporophyte generation is this yellow-like, stalk-like structure with a capsule there on the end. And it's in the capsule where the spores are produced. The sporophyte, remember, that generation, all the cells are diploid, and the gametophyte, these are all haploid. So let's take a look here. You're actually going to have separate male and female plants. This is, on one side is the male, this is the female. These plants don't get very big. They cannot because they don't have vascular tissue. And it does look very plant-like. Their growth pattern, unlike the liverworts, is a stem-like structure, and the leaves, leaf-like little leaflets come out and go around in a radial fashion, so sort of radial symmetry to their plant body. And up on the top is where you're going to have your gametangia. If this is male, then that's your antheridium. Okay. So up here, this entire structure on the top would be called the antheridial head. And that's where you find your antheridia. So they're showing you, we're zooming in to look within the antheridial head, there's your antheridia, and within these organs here is, remember, the body of the plant is already haploid, all of the cells. So if we're going to make sperm, it's got to be mitosis, not meiosis, and differentiation. So the sperm are produced in there, and just like with the liverworts, the earlier group, if a drop of water smashes on top of that head there, it can scatter the sperm onto a neighboring female, and then the sperm swim into the archegonia. The female has an archegonial head. And on the archegonial head, you're going to have you can have several archegonia, and there, that little blue structure in there is a haploid egg. Remember, the body again is all haploid, so in order to make the egg, a special cell in there is going to divide by mitosis and then go through differentiation, switch on a different set of genes so that it behaves like an egg. So let's say we follow one of these archegonia and a sperm makes it in there and fertilizes the egg. At fertilization, this egg is diploid. It's now diploid, and it's no longer an egg. It's called a zygote because the sperm went inside the egg and the two nuclei fused together. Now that cell within an archegonia, which is an archegonial head, which is still attached to the rest of the female gametophyte, is going to begin to develop. It's going to go through cell division, forming an early embryo. All of the cells that are in blue, they color it blue so we can distinguish, are diploid. The cells of the archegonium, which is part of the female gametophyte, those are haploid. Okay, so the gametophyte begins to grow out of the diploid sporophyte begins to grow out of the haploid gametophyte and eventually turns into this stalk here and a capsule. And in the capsule is where you're going to have the sporangium. Sporangium would be where the spores are produced. The stalk has a fancy name you'll see in the lab manual called a seta. S-E-T-A. Setae plural if you put an E at the end to follow the rules of pluralizing in the old languages. Within that capsule are going to be the spore mother cells. And the spore mother cells are diploid just like any other. Another name for spore mother cells are sporocytes. The sporocytes are the ones that they're diploid and they go through meiosis. So within this capsule, in there are special cells that have differentiated to go through meiosis, and they're called spore mother cells or sporocytes. Those cells that are within there go through meiosis, and then the spores are released during the right time, and they get scattered, and when they land, they start to germinate, and some are going to germinate to form a female, and some germinate to form a male, and we've completed the life cycle. Eventually, you get the next generation sporophyte, which is all diploid. So there you see an alternation of generations. You're going to be expected to interpret these, and so knowing the terminology is very important. So it could be a question with just words and no picture. It could be a picture with labels removed and asking you, what is the name of this structure? Is it the sporophyte? Is it the gametophyte? What process occurs? Anytime you go, it's this simple. Anytime you go from diploid to haploid, you have to be meiosis. You're reducing the number of chromosomes. Anytime you go from haploid over here to diploid on this side here, fertilization occurs. The two gametes unite. Okay, so those are the major processes that occur. Everywhere else, mitosis occurs as these things grow and develop, and differentiation. Everywhere else. Any other arrow of development, mitosis and differentiation. And then we end with the hornworts and anthocerophyte as a reference to that, the horn-like structures that grow out of them, like the liverworts, not like the mosses we just finished covering. The liverworts, going back to the hepatophytes. These guys have a flattened body that occurs on the substrate that we're growing on. They'll have little rhizoids that anchor into the ground. And the gametophyte, which comes from a fertilized egg, just like with the prior group, the mosses, the Bryophyta. It ends up growing a stalk, just like we saw earlier, but and so this right here, it looks like a horn. That horn-like structure is your, it's your sporophyte. The body of the plant here is the gametophyte. Okay. And they don't grow a capsule, but within the stalk there that looks like a horn or horn-like structure, that's where the sporangia is, and that's where the spores are produced by meiosis. Remember, the sporophyte is diploid. The rest of the body here is haploid. Okay. Now, how did we get the sporophyte? Well, sperm had to go and fertilize an egg, and that zygote develops into this stalk here, which is the sporophyte generation. So here the sporophyte totally depends on the gametophyte. Not completely true here in total dependence, because you can see, unlike with the mosses earlier, the sporophytes are not green. It means they're not photosynthetic. So they're totally depending on the gametophyte that they're growing out of for their nourishment. Because they can't make their own food. So again, I mentioned there's some exceptions to the rule here. The sporophyte does depend on the gametophyte. It has to grow out of there. The early growth of the embryo before it becomes this mature sporophyte is protected by the gametophyte. But these guys here are green and they're photosynthetic too. So both the sporophyte and the gametophyte are photosynthetic in the Anthocerophyta. Now, as a group, the hornworts, they're commonly called. Their origin is puzzling because they don't find any spores in the fossil record until the Cretaceous, whereas those for the other non-tracheophytes, which are we call them overall bryophytes, and then there's the formal Bryophyta, formal taxon. But generally, they all evolved much earlier. Okay, so you go back in time here, and you find this ancestor right here, but they don't find any spores in the fossils until the Cretaceous, when the angiosperms have already evolved. The angiosperms don't evolve until whatever time that is right there. Okay, and that's sometime within the Cretaceous period, which is much later than when these early plants evolved. Okay, so again, the Sporophyte is actually photosynthetic for the Anthocerophyta, but the Sporophyte is still dependent on the Gametophyte. It still has to grow out of the Gametophyte, and so that's the last group. Now, going and looking at the cladogram more closely here, we can see that there's your liverworts, which were the Hepatophyta or Hepatocophyta. Here's the phylum Bryophyta, and this is the phylum Anthocerophyta, which we just covered here. And now we're going to be moving on to the vascular plants, which we call the Tracheophytes. So in this section here, we are going to be looking at the Tracheophytes, and we were looking at the non-Tracheophytes. The non-Tracheophytes were avascular, non-vascular. A means without. So this is another way of saying the vascular plants. They call them Tracheophytes because they possess the xylem that would have those special cell types called tracheids. And a big thing to remember here is now we've shifted from the Gametophyte generation dominating. Now the Sporophyte or the diploid generation dominates here. And so your learning objectives or your learning outcomes for this one are to explain the evolutionary significance of tracheids, which allow for efficient water conduction up the plant, and then evaluate the significance of roots, stems, and leaves in the Tracheophytes. Why are those structures important in their evolution? Now, what you're looking at here, and they show like a sewing needle next to a fossil, and here's what the fossil looks like. You can see this branching stem-like structure here. This is one of the earliest fossils of vascular plant or a tracheophyte. They named it, they classified it in a genus called Cooksonia, and in a phylum that's now extinct. So this phylum of plants is a complete branch on the evolutionary tree has gone extinct. None of these exist anymore. This genus Cooksonia is classified in a phylum called Rhinophyta. And this we see in fossils about 420 million years old, where the first, which will mark the time about which the first tracheophytes evolved. Was not very big. You can see we have sporangia here. We already know from earlier that sporangia is where spores are produced by meiosis, and that tells me something here, that the body of this plant is diploid, and because it has sporangia growing on it, it is the sporophyte. And so the cells that make up the sporangia are also diploid, and in here spores would be produced by meiosis, haploid spores. You're going to see some terms now that we're going to start to use: homosporous, and the alternate would be heterosporous. This means when you're homosporous, you produce one kind of spore. When we start looking at more advanced plants, we get to the seed-making plants, they're going to produce two kinds of spores, a large spore and a small spore. But for now, the plants we're looking at are still making one kind of spore, homospores. Okay. And so a key thing here is the evolution of vascular tissue. We did not see that in the bryophytes in the last section. And two major types of tissue found in the vascular tissues include xylem and phloem. And xylem is basically going to be conducting water and minerals upward, up to the leaves and the plants above, the shoots above the ground. And the phloem basically is going to conduct organic molecules like the sugars produced by photosynthesis. Glucose gets turned into sucrose, which is our table sugar, or what we use to mix in tea and other things. Hormones, amino acids, anything organic produced by living cells of the plants gets conducted back down and around the plant. So if we're going up to the leaves with the xylem, we come back down out of the leaves to other parts, including the roots of the plant. So if we look at a cross section through a stem here, you can see some of that vascular tissue is in brown and orange. The orange is the xylem, is typically going to be found in some of the plants we're going to be studying here toward the inside of the stem, and towards the outside would be the phloem. The xylem, which is seen here enlarged, is made of these special cells that we might call tracheids and vessels, or tracheids gets its name from. There's some distinction between the kinds of cells that you find in there. The key, though, is that when these cells mature, they're dead. They just have the cell wall left behind, and that's good enough to conduct water up. So the water and the minerals come up here, go to the leaves, and then in the leaves, the water gets pulled back in and then comes back down the phloem. And the phloem is made of living cells here. So the phloem would be made of those living cells there. And then this is a cross-section of an actual stem that's been stained. And you can see these bundles of vascular tissue that actually run up and down the stem there. And you'll study more of that plant structure both in chapter 35 when we get there in the lecture, and in the laboratory. There is a lab on this plant structures overall. So what is the key thing about vascular tissue is now this is another step of moving away from water more fully. The Bryophytes didn't have vascular tissue, and that forces them to be relatively small and many times having to grow near water overall. A key thing here is going to be something to remember is that now that the Sporophyte generation is dominant, this is going to be the larger plant overall, that you find the vascular tissue in the Sporophyte. This right here, this statement's profound here. It's going to be important to remember. It's a big idea. The vascular tissue, which is this slide is about, develops in the Sporophyte. You're not going to see vascular tissue in the Gametophyte. General rule to follow. Okay. Other things that are important: cuticle, which is a waxy covering, and stomata. Those are also important adaptations to moving on to land, which was discussed in an earlier section. Remember, the cuticle is going to prevent water loss across the surface, and then the stomata are the little openings. It literally translates to mouth. Stoma means mouth. These little guard cells help control opening and closing. The plant needs to exchange carbon dioxide and oxygen, but if it's too dry, we need to close that up to prevent water loss through those little openings, those little stoma. So a tracheophyte, again, means vascular plant. So that's what this translates to. You should just make sure you know that as a matter of knowing. Vascular plant has the vascular tissues. So the vascular plant's going to include 7 phyla. Extant means still living. Other phyla have gone extinct, like that Rhiniophyta for that fossil. Okay. And there's 3 big groups. There's the Lycophytes, and the actual name is Lycophyta. Those are the club mosses. The Pterophyta, or Pterophytes, these are the ferns, whisk ferns, and horsetails. And then the seed plants, which we sometimes called Spermatophytes. Remember, phyte means plant, and sperm means seed. So this literally translates to the seed plants. Okay, so again, we're going to see this trend like we saw in this image earlier. We're going to start to see a reduction in the Gametophyte and the dominance of these Sporophytes. We're not going to cover these guys in this section because these 2 right here, those are your Spermatophytes. So we're not looking at seed plants yet. That's the next chapter. Okay. But here again, we're going to see the Gametophyte starts to become reduced and the Sporophyte becomes the dominant generation that evolves the vascular tissue. Now, when the Gametophyte gets smaller, the gametangia, where the gametes are produced, also becomes smaller. So you're going to see that similar reduction. Smaller Gametophyte, smaller generation. Then the gametangia, where the gamete is produced, also has to be smaller to be part of that smaller generation. So these are the two major groups we're going to be looking at right now, going off of here. The seed plants, which are these right here, are going to be the substance of the next chapter. Right now we're going to focus on these vascular plants that are seedless. So these are seedless vascular plants in this part of the chapter. We still don't have seeds. What we have now is vascular tissue. The club mosses belong to a formal taxon called the phylum Lycophyta or Lycophyta. They're homosporous. Their sperm are motile with flagella, and anytime you have sperm with flagella, you're going to need water. So these guys still need water to complete their life cycle, these plants. Okay. One phylum here called the Pterophyta, the P is silent. Make sure you check on spelling that because you might be asked to spell it on a lab exam. And they include the ferns, horsetails, and whisk ferns. Okay. When I was learning the taxonomy, probably more than 20 years ago, these were actually different phyla. They've grouped them together now to fit better their evolutionary history. Keep in mind when you read the descriptions here, homosporous. Few are heterosporous. We're not going to focus too much on the heterospory in ferns, but there's a group called the water ferns that are heterosporous. This is mentioned a little bit more in your lab manual overall. It gives some other stats in there, like there's how many genera, how many species. Each one, don't memorize how many species, but be aware, pretty abundant, the ferns overall. The horsetails, homosporous, no heterospory, motile sperms, so you need water for the life cycle. And for the whisk ferns, same thing, homosporous, sperms have flagella, so you need external water here. Keep in mind, check out the number of living species today, about 15 identified. And here for the whisk ferns, only about 6. Okay, so you don't necessarily memorize the number, but just have that idea that when you look at horsetails, not very many species alive today, and whisk ferns, not very many. Now that we have true vascular tissue, now we can define true stems, roots, and leaves, but they had to evolve. These are organs that had to evolve as vascular tissue is evolving. So the first structure to actually evolve in the fossils, like we saw with Cooksonia, was stems. So you had vascular plants that had stems, but no roots or leaves. Okay, so stems evolved first. Okay. And because these early plants lacked roots, that limited what those early tracheophytes could do, just how big they can grow, how much stuff they can pull from the ground, like water and minerals and other nutrients they need to grow. So again, stems evolve first. Okay, so no roots or leaves yet. Okay, what are roots? Roots are organs that evolve later, okay, and they have to have that xylem and phloem in there as well. And so this is going to allow for more efficient transport, and roots help anchor and support your plants. So now you can get even bigger above the ground. And it turns out that the roots that evolve in any other vascular plant besides the club mosses, the Lycophyta, which I mentioned just a while ago, the Lycophyta diverged early on the evolutionary tree before the roots had evolved. So the Lycophytes, the club mosses, evolved their own roots independent of the other vascular plants. Okay, so the Lycophytes, the ferns, the Spermatophytes, the seed ones that we're gonna study later, that evolve later, they evolved their roots from common ancestor. The Lycophytes evolved it from another ancestor. They just two independent. They evolved the same type of structure independently of each other. Kind of like the discussion of how the whale and the shark end up having the same shape. They evolved the same type of similar structure because of the same kinds of pressures from the environment. And then the leaves evolve. Okay, and what do leaves do? The main function there is to capture sunlight for photosynthesis. They have to have vascular tissue in there. And again, the leaves evolve after the stems do, and the Lycophytes, the Lycophyta, the club mosses, evolve their leaves independent of the rest of the vascular plants. So if you're the rest of the vascular plants, we're going to call these true leaves, just like eukaryote means true nucleus, the root of U. And then this part right here, PYLL, means leaf, okay, in translation. So anytime you see mesophyll, euphyll, microphyll, lycophyll, when you learn that root, you learn a lot of terms, because you see it as a root in the word here. So the euphylls are found in the other plants, and sometimes they refer to them as megaphylls, which means large leaf. Now, some megaphylls can be quite small, but they're nothing like the lycophylls. The lycophylls are found in lycophytes, and they have a much simpler structure, and they're by the way they're structured, they can't get big at all. There's no potential for them to get large. So sometimes they refer to lycophylls as well as being microphylls. Okay, so be aware that microphyll and lycophyll synonyms. Megaphyll and euphyll synonyms. So here is in the lycophytes. So in the line or the branch of the evolutionary tree that gave rise to the phylum Lycophyta. This is a proposal of how those leaves might have evolved. We know that the early vascular plants just started off with a stem. Okay? And that stem had to have vascular tissue, which is in dark green in the middle there. Okay? So there's your vascular tissue. So eventually, it seems that perhaps little fleshy outgrowths, leafy-like tissue, began to grow out of the stem. Capture more sunlight that way, more surface area to do that. And then eventually the vascular tissue starts to grow inside that little leafy tissue, forming a very primitive leaf, a lycophyll. And then eventually you get your modern form of the lycophyll, the microphyll. And what limits the microphyll is it's only going to have one vascular strand or one strand of vascular tissue in there, which we call veins in the leaves. Like the veins we have in our body, except these are veins that are in plants. Okay? So they have only one vein, and that limits how big they can get. So that's why sometimes they refer to these as microphylls. Okay. And then for the origin of the true leaves, the megaphyll, the true leaves or the megaphylls as well. If we go back to the way Cooksonia looked, that first picture in this section, Cooksonia was just a branch and a bunch of stems. They even had some sporangia there at the ends there. So you can imagine a branching plant body, some branches coming towards you in my picture, some going away from you back behind the plane of the screen you're looking at, and some going forward. So here you just have branch with vascular tissue, branching. Then you get, and the branching is roughly equal. Then the proposal would be that all of a sudden you start to get unequal branching. The branches don't grow all evenly. And then the branches now go on a single plane. So those branches that I drew coming towards us and behind the plane of the screen, now they go away and you just have branches on one plane. Okay? And then those branches don't have any leafy tissue around there. Eventually leafy tissue evolves to grow around there, and now you have your primitive true leaf. Eventually you get a true leaf, a euphyll or a megaphyll that has many vascular strands or veins, not just one vein. And so that's the difference between a lycophyll and a euphyll, is many branches of veins within there. And so ultimately, that's about what we need for this chapter here, because we're going to end it with just the tracheophytes that are seedless. But in the next chapter, we're going to cover seeds, and that's something else that's going to eventually evolve later on. And the seed is going to be highly resistant when that embryo is retained. Here's the embryo. This is a bean seed, which is a kind of seed plant. There's the embryo, and it's packed inside this protective coating. That protective coating is called a seed coat, and it prevents the embryo from drying out. And it's also going to be packed with food, kind of like an egg does, where the egg has yolk in it. That food is called the endosperm. Now, this is a corn seed, and the corn seed, the endosperm kind of stays separate from the rest of the plant, but here, the embryo is going to have two structures called cotyledons that absorbs that endosperm. So the endosperm is within those cotyledons. So your food source is here and here, which will nourish that embryo when the embryo begins to germinate. And that embryo, carrying that food supply within that seed, will not germinate unless water begins to penetrate the seed coat. So the seed coat has to have water penetrate inward, and once water penetrates inward, first it's water, then water is going to allow oxygen to penetrate inward. Once oxygen hits the embryo, that starts the metabolism. So when you're starting your metabolism, that's kind of the reference to what germination means. So right now, In a seed that's not germinating, the embryo is in a suspended animation. Its metabolism is zero. It's not exchanging oxygen. It's not producing carbon dioxide. Okay, so keep in mind we're going to be looking at Lycophytes and Pterophytes in this chapter, and they do not have seeds. The seeds come later. Okay, that's the next chapter, but that's an important development later on. And then not just seeds, but later on you're going to have fruits and that develop from flowering plants, the ultimate seed plant that we'll study later called angiosperms. So that ends up packing a seed inside of a fruit, and that fruit actually helps the seed disperse with the embryo, move it further away from the parent plant. And these seeds may be moved away by animals, which are trying to use the seed for a food source, or the wind might carry these seeds away, depends on what kind of seeds you have. So looking now at the evolution of plants, we started out here with the Charophytes, the closest representatives here. We go back further, we have some ancestral algae, green algae, and here's your chlorophytes here. Well, the first thing we start to see is chlorophyll A and B, which we would find in the chloroplast, which remember evolved from endosymbiosis. They used to be cyanobacteria. So cyanobacteria gets taken into an early protist ancestral and then becomes the algae, the green algae. We still have green algae today. And then plasmodesmata evolves. These are those shared derived characters that we talked about in chapter 23 when we're talking about systematics and cladistics. So here, this ancestor has plasmodesmata evolved. Plasmodesmata was something covered in the chapter on cells back in your Biology 1 course. Plant cells tend to be rectangular in shape, and they have a cell wall. But the cell walls have these little pits in them, or little holes, that allow stuff to move from the cytoplasm of one cell to another. Those little pits, or the little holes within the cell wall, are called plasmodesmata. That's important. You can move materials back and forth. Then this ancestor evolves the cuticle, the antheridia, and the archegonia, which is where sperm and eggs are produced, and your embryo retention. The multicellular embryo is retained in all of these plants that are connected to this common ancestor. That includes the liverworts all the way to the angiosperms. And then stomata evolve here. Stomata allows the plant to exchange carbon dioxide and oxygen. Stomata evolve here, and then they didn't indicate what a derived character was there, but there's some common ancestor here between the hornworts and the vascular plants. So over here now is your ancestor to all vascular plants. Cooksonia is one of those early plants. Cooksonia may not be the common ancestor. That's not the assumption, is that there is an ancestor that started out to have vascular tissue. Our Sporophyte is now dominant, and now you have true stems, roots, and leaves. And over here we have our megaphils, our euphils, and over here we have our lycophils that evolve independently. Also the roots, a different kind of root evolves in this branch than over here from a different ancestor. And then over here we have our seeds evolve. These are this common ancestor here is the common ancestor to the spermatophytes. So this section is on the phytum Lycophyta, which we mentioned earlier section that this group diverged or evolved independently from the main other group of vascular plants. They evolved independently their own leaves and their own roots. And remember, they have the lycophils or the microphils, which are the small leaves. This group, for this group, your learning outcomes or objectives are to explain the features that differentiate the lycophytes, which is this group, from the bryophytes, which are the mosses, liverworts, and hornworts. The Lycophyta are sometimes referred to as club mosses. And so when we look at that comparison there, they kind of resemble mosses in some ways, except they have vascular tissue, so the resemblance is there. They don't produce seeds. They require water to complete their life cycle, but again, they have vascular tissue, so that distinguishes them from true mosses. The other thing is that if you look at the fossil record, Lycophytes were among some of the dominant plants. You go back to the Carboniferous period, which is a time in which the plant life that was on land at that time, when died, got buried, ended up being their bodies ended up being turned into coal. So the coal that we get from there includes some members of the Lycophyte that are now extinct that were actually really large. So there were some large Lycophytes early on. They've just gotten extinct now. And so, again, they superficially resemble those true mosses. The Sporophyte is dominant. That's another difference between true mosses and Lycophytes. The Lycophytes do produce a Gametophyte, and the Gametophyte is smaller and photosynthetic. Okay, so that's different than true mosses. In true mosses, the Gametophyte was dominant. Now, the Gametophyte is photosynthetic, but the Sporophyte was totally dependent on the Gametophyte. Here, there's not that much dependence on each one, but the more conspicuous is this Sporophyte generation here. And there on the little ends there, you can see the little sporangia, which we can think of as little modified leaves, leaf-like structures where the sporangia now occur. So how are these guys different from other vascular plants? Well, one, they're not as large as other vascular plants, and you can look at other vascular plants that include the spermatophytes that produce seeds. These guys do not produce seeds; they lack seeds. Although we're going to look at ferns right now, and they don't have seeds as well. So that takes care of your second learning objective. So the book does discuss these comparisons a little further. But this is an important group, and remember, this is that group that it diverged away from the rest of the ones, including the ferns and their allies, like the whisk ferns and the spermatophytes or the seed plants. The Pterophytes are a group of plants that include the ferns and their close relatives, and formally they belong to the taxon, the phylum Pterophyta. And so make sure you're familiar with that term. Now, the learning outcomes for this section are to list the features that we see in Pterophytes and contrast Pterophyte and moss Sporophytes. So how are they different from the vascular mosses? So some more recent studies have found that that understanding the relationship with this group is still not well understood. But generally, it's been agreed that there are two major clades, and this includes the whisk fern clade and the horsetail clade. And both do include ferns that we see there. And here's your horsetail and your whisk ferns, which we're going to briefly look at these groups here and then look at the ferns overall. And then you can see where these are relative to the Lycophytes, which we just studied here. And then up next on the next chapter is the seed plants. Now, when it comes to the Pterophytes, the ferns and their allies or their relatives, they do have antheridia and archegonia. Remember, antheridia is going to be the structure where sperm are produced, and archegonia where eggs are produced. And these are going to be structures or organs of the Gametophyte generation. So let's say the Gametophyte generation. So this is not new. This is something that we cover as a generalized type of thing. Remember, the Gametophytes produce the gametes, and they produce the gametes by mitosis and differentiation, and the Sporophytes would produce the spores. Now these ferns, although they have vascular tissue, just like Lycophyta in the prior section, they're still going to require water to complete their life cycle because their sperm are flagellated. They have flagella, and they need to be able to swim to fertilize the egg. So water, we still haven't broke free from water. Sometimes I like to compare these to the amphibians. They require water to complete their life cycle. Though amphibians are an animal, and these are plants, right? So here are the whisk ferns, and they kind of look like a whisk, like kind of a brush or a broom type structure here. And these remind me of that fossil Tracheophyte from 400 and some million years ago, where you have that Cooksonia fossil. These whisk ferns are found in the tropics. They don't have any leaves, true leaves, and they don't have any roots, just like we see some of the most primitive vascular plants first evolved the stems first, were the first ones to evolve. Then it was leaves, then it was plants, then roots. You can see these little yellow structures there on the end. Those are the sporangia, and what we're looking at is the dominant sporophyte generation. The sporophyte has the sporangia that produce the spores by meiosis. Okay, so these yellow structures here are the sporangia. Okay. So these are the whisk ferns, and they're in the group Pterophyta. Now, they're gametophytes. This is interesting. It's the sporophyte that always has the vascular tissue. I mentioned that in the earlier chapter, but some gametophytes within this group do develop some rudimentary vascular tissue, but nothing major. So we can still consider it that vascular tissue is typical of the sporophyte generation. Okay. So it's an exception, small exception, and it's still basic vascular. It's not even like true vascular tissue. For the horsetails, there's 15 extant or living species. We went over whether they were homosporous. Most of them that we've been talking about are homosporous. The ferns there's water ferns that have some heterospory, but for the most part, the Lycophytes, the club mosses, these horsetails here and the whisk ferns, they're homosporous pretty much. There is only one genus though. Okay, we don't have several genera. It's just Equisetum. And when I think about horses, I think about equine. That root in there means horse, although this is not an animal, but it must be that the structure of the body of this plant reminded somebody of a horse's tail, perhaps. This is why they're called horsetails. Now the sporophyte, which is what we're looking at, is the dominant generation. It's going to have these nodes or rib-like structure. You can see the nodes running up the plant. It looks like it has little whorls of little leaf-like type structures there. But for the most part, the stem itself can do photosynthesis. You can see how the stems look green there. And then on the ends here, on the tips, we have what it almost looks like a pine cone. And by the way, the Lycophyta produce a structure like this too, and they call these structures strobilus, or strobili plural. And the strobilus, each one of those little structures in there contains a sporangium. And the way the plant biology describes these are thought to be modified leaves that grow sporangia on them as part of the body of the plant. So the spores are produced in here. This is the sporophyte. Okay. Everything you see there is a sporophyte generation. Okay, so that means they're all diploid cells, even within the strobilus, all of the cells there. But within there, you're going to have your spore mother cells, the sporocytes, that go through meiosis to produce your haploid spores. They go and they produce the gametophyte generation, which is much reduced here. They're not showing the life cycles, but it's the same concept over and over again. The sporophyte produces the haploid spores by meiosis. Okay, now these also have the nickname or a common name of scouring rush because their cells do have glass deposited within their cell walls. And the pioneers back in those days used to use these to clean off pots and pans and stuff to scour them, much like we might use steel wool to clean as a rough surface to clean things. So that's how they get their name. Moving on to ferns. The ferns are the most abundant seedless vascular plants. Okay, so these are without seeds, but vascular. You can see that today there's still a very large fern species called a tree fern. And it can get many, many meters up into the air there, but it still has the characteristics of ferns. And most ferns are much smaller than this, and they're not as small as club mosses and so on, but they're relatively tall, or the horsetails. Those are all relatively short in stature plants. But ferns used to, just like the Lycophytes, used to have lots of species that were large like this. They dominated forests back then. Angiosperms were not dominating in those times. The flowering plants have not really evolved and fully become dominant. And here, so these guys, along with the Lycophytes, are the ones responsible for forming coal when you go back to the Carboniferous period, then during that time, geological time period. The Sporophyte, as we've been talking about, is going to be the one that's conspicuous, and the Gametophyte is much smaller, much reduced. And here, both of them are photosynthetic. So we're going to go over their life cycle a little bit. But let me show you here. This is the life cycle involves a Gametophyte and then the familiar fern plant. You see ferns sold a lot in garden centers. But here, this is the entire Gametophyte generation. And if I had to put a size coming from here to here, this is about no bigger than 5 millimeters across. Those little itty bitty lines on a metric ruler. These are very tiny. This is the entire Gametophyte generation. The Gametophyte lacks vascular tissue. It's very small. We're actually looking at the underside of it, and it has these little rhizoids, just like mosses do. Just like mosses, they don't have, their Gametophyte generation doesn't have vascular tissue overall. And then this particular Gametophyte is carrying both the male and the female organs that produce the sperm and the egg. The archegonia are right here, and here the antheridion. The idea here is to have the two develop at different times so that we don't self-fertilize. It's going to create less diversity if you fertilize your own eggs. There'll still be some diversity because of well, I'm now getting into confusing with plants, so I'll scratch what I'm saying. The idea here is that they're not going to develop at the same time, so the antheridium may develop first, and then a neighboring Gametophyte may be at a slightly different stage of development. But you still need water present, so if the gametangia don't develop at the same time, then the antheridia can develop and fall off, release the sperm, and then the sperm can't fertilize their own eggs because the archegonia hasn't fully developed. So the sperm, however they might move about, they require water, can go and fertilize a neighboring Gametophyte's eggs where the other one had developed further along. So there's this asynchronous development within these populations to promote cross-fertilization. Here, the Sporophyte is going to be dominant. The Sporophyte actually depends on the Gametophyte for a small period of time, and I'll show you that in the life cycle right here. So this is supposed to represent the Gametophyte generation, and here all of the cells are haploid. We're on the above the line here in this beige or peach-colored area there. They have the archegonia here, which are located near this cleft area right here in the body of the plant. And we're looking at the underside of this Gametophyte. It's very small. Remember, this thing is not much more than 0.5 centimeters or 5 millimeters across. They're your little rhizoids that grow into the ground. And so within here, all your cells are haploid. Sperm are produced by mitosis and division, differentiation, and then the sperm go and fertilize an egg, ideally in the archegonia of another Gametophyte, neighboring probably. Water is required because the sperm need to swim. So the sperm do swim into the archegonia, fertilize the egg. Remember, when we fertilize the egg, you're first going to get the zygote, and the zygote is going to be retained within the archegonium. So the zygote would be retained within this on the body of the Gametophyte. The zygote begins to divide, forming our embryo, and the embryo continues to grow out of this Gametophyte. So as it's starting to grow out of there, it's growing from the underside, then begins to move upwards through that little cleft area there, and begins to grow upward. And while these are your little rhizoids from the body of the Gametophyte, here you can see a true root beginning to form from this Sporophyte. Okay. Eventually, this Sporophyte gets big enough, and the Gametophyte dies and degenerates, and this root begins to grow an underground stem called a rhizome. So that's a plant anatomy term. A rhizome it's actually a stem. It's not a root, but it's underground, and it grows horizontal underground. So it's an underground, not root, but stem. And leaves grow from stems. So in this case, the leaves are going to begin growing as these little immature young leaves, they have the nickname fiddlehead because they look like the end of a violin. So these little fiddleheads begin growing, and they uncoil and end up growing into larger and larger leaves, which the leaves are called fronds. A frond is just a fancy word for the leaf. Like if I say the fronds on a palm tree, the palm tree is in the seed plants, but frond just means leaf, right? So these little fiddleheads sprout up from little nodes along the rhizome. Eventually they mature. So here's your mature frond. It looks somewhat feathery, typical of fern. Now, fern leaves, but there's variation in what their leaves look like. But all of these ferns, underneath them, the fronds, they're megaphylls. They're euphylls. They're true leaves, right? But sometimes they refer to the fronds that grow underneath the sporangia. They'll call them sporophytes. You're going to see that term thrown out at you in the lab in the lab manual. So what is a sporophyte? Remember this root right here, phyll means leaf. There's going to be a leaf that houses or holds a sporangium. So the sporangia are going to grow in clusters under the leaf here. Okay? I'll show you a picture of what one looks like more the entire leaf underneath. But let's say I took a cross-section through a leaf, and this is the cross-section right here. Okay? This is inside the leaf. This is the mesophyll, which means the inside of the leaf. Hanging from underneath it is a sorus, or sori plural. The sorus is a little structure that has a cluster of sporangia. So there is the sorus on your diagram from your book, and then it says cluster of sporangia there. Okay. So each one of those little ball-like structures within the sorus is a sporangium. So now they've removed one of those sporangia, and inside, remember, what do you have in there? The entire body of this plant now is now diploid because of that fertilization. So the cells that make up the sporangia are also diploid. But there's going to be special cells in there called mother cell sporocytes. Spore mother cells. They're also called sporocytes. That's the synonym. Okay. So your sporocytes within there, which are the spore mother cells, those are the ones that are going to go through meiosis. Okay. And each one of those sporocytes divides twice to make 4 cells that become spores. The spores get scattered. They're all haploid now. They get scattered, they germinate, they begin to grow the next generation of your gametophyte. All of the cells there are haploid. Okay? So that's your generation there, and there you see the alternation of generations. And then we repeat. Your antheridium and your archegonia produce your sperm and your eggs, respectively. So a little bit of fern morphology. I already mentioned the idea that the rhizomes, those are underground stems, right? So you got your underground stem below the ground, and then you're going to have your little fiddleheads growing there, and then the leaves begin to mature more, and then you have your mature fern frond. Okay. And these are examples of some coiled up fiddleheads from some different species. This is what they look like. So those are the young fiddleheads. And then this would be the rhizome. And this would be a mature frond. So when I was mentioning the frond, this is what the frond of a typical fern would look like here on the top. And we're looking under a frond here. This entire thing is the frond. It has little the entire leaf itself is cut up into smaller sections or little leaflets. And underneath each one of those little orange ball-like structures is a sorus. Plural is sori, which I mentioned earlier. So they're underneath the fronds. So we could refer to this leaf here as a sporophyll because it has the sporangia underneath it, right? Now, if I were to section through one of those, this is what the image of a section through a sorus looks like. And we can compare this to the diagram earlier. This is the inside of the leaf here, the mesophyll. This is the structure. It's a little shield-like structure in which you find the sporangia there. Okay, so this entire structure here is called the sorus, and then the shield has a name called there's anatomy here to this, right? But each one of these little circular structures here is a sporangium where meiosis occurs to produce the spores. Okay? Now, remember that the gametophyte, okay, when the spores are released, they germinate, they're going to form a gametophyte, which is a little heart-shaped gametophyte. They actually have a name for this, the body of your gametophyte as well. That'll be brought up in the lab manual. It's going to have this little rhizoids, no vascular tissue, and the sperm produced in the antheridia are flagellated, so they need water to complete the life cycle.